Welcome to the deep dive. Think of knowledge not just as static facts, but as this incredible story constantly unfolding.
That's a great way to put it.
And today we're focusing on a really amazing chapter, the history written deep inside our own DNA and, well, the DNA of all life, really. Here on the All Sorts podcast, we really believe that understanding the past, even the really, really deep past, gives us such vital context for, everything happening now. So we're taking a deep dive into the stack of sources all about the, frankly, revolutionary field of ancient DNA research.
That's moving so fast.
It really is. Think of this as your shortcut to getting properly up to speed on what scientists are discovering, the tech that makes it all possible, and of course the big challenges they're still grappling with.
It is complex, and it's definitely rewriting the history books in some areas.
For sure. All Sorts podcast is your guide today as we navigate this whole fascinating landscape. And hey, if unlocking these hidden stories, ancient human journeys, figuring out extinct animals, how our ancestors adapted, if that gets you excited, and honestly, it should.
It's incredible stuff.
Then you've definitely found your people. The best way to keep exploring these kinds of mind-blowing topics with us to catch all our future deep dives is to hit like, share this, and subscribe to the All Sorts podcast over on YouTube.
Definitely do that.
And come find us on X, Instagram, and Threads, too. We've already covered so many cool subjects and other deep dives. There's just a whole universe of knowledge waiting for you there. Okay, let's get into it. Today's mission. Unpack what these sources tell us about ancient DNA research, sometimes called paleogenomics.
Paleogenomics or archaeogenomics, yeah. Same idea.
Right. We want to explore what it is, the technology driving it, the big hurdles, and the amazing insights it's giving us. Insights into human migration, species evolution, even ancient diseases and farming.
It's a huge range. From extinct megafauna right down to, well, potatoes, as we'll see.
Potatoes. Okay, I'm intrigued. So first off, what is this field, fundamentally?
At its core, it's about finding, extracting, and then analyzing DNA from old biological stuff. Bones, teeth, soil, whatever holds ancient genetic material.
Okay.
And the sources are really clear. The number of studies using ancient DNA or aDNA, it's just skyrocketed.
Right. It's gone from being this really niche, almost sci-fi thing to a major scientific tool.
Absolutely. A real engine of discovery now.
And it's not just about human history, is it? The sources say a pretty straightforward use is connecting extinct animals to living ones.
Exactly. That's one of the clearest paths. You use the ancient DNA to build these molecular phylogenies. Think of them like really detailed genetic family trees. They let you place long extinct species right onto the evolutionary map with their modern cousins.
That's so cool. So instead of just comparing bone shapes, you're reading the actual genetic instructions.
Precisely. It gets past some of the ambiguities of just looking at physical form.
What are some examples the sources mentioned?
Oh, there were several good ones. They talked about using aDNA on the Australian marsupial wolf, the thylacine.
Right.
Also, New Zealand moas, those giant extinct birds. American ground sloths, too, and even some endemic Hawaiian geese. Basically, animals we knew from fossils or history, now we can pin down their genetic relatives.
Which must mean places like museums with all their old collections are suddenly incredibly valuable genetic archives.
Totally. Museums are becoming critical genetic repositories. Some are even setting up their own specialized labs, creating standards for how to sample these precious specimens without destroying them or contaminating the DNA.
But getting usable DNA isn't always straightforward, right? Especially, the sources mentioned, from certain environments or for studying things like non-human primates.
Yeah, that's a huge factor. DNA preservation is really variable. It depends massively on the conditions after death.
Like temperature and humidity.
Exactly. Hot, humid places, especially with acidic soil. Think tropical rainforests. They're just terrible for DNA survival. And since a lot of non-human primates lived and live in those kinds of environments, studying their ancient DNA is, well, tough.
So climate really dictates what's possible. But even with those difficulties, they are making progress, right? Like with those extinct lemurs in Madagascar.
That's a perfect example. They managed to get ancient mitochondrial DNA, which often preserves a bit better than the DNA from the cell nucleus from these recently extinct lemurs.
Okay.
And what they found actually overturned some of the previous classifications that were based just on the lemur's physical shape, their morphology. Their genetic relationships were different than we thought just from looking at them.
Wow.
And this kind of work, it's also opening doors to understanding the history and extinction of other species, like macaques that used to live in Europe or ancient apes in Asia during the late Pleistocene. Stuff we only had fossils for before.
Okay, so let's talk about why this field is booming now. It's not just that scientists got more curious, right? The sources point to specific tech breakthroughs.
Oh, absolutely. That's the main driver. Huge improvements in how we extract DNA from ancient samples and how we sequence it, read the genetic code. Modern extraction methods have become super efficient. One source described it as almost a factory-like method, where labs can process hundreds of specimens pretty rapidly now.
Hundreds at once. That's a total game changer compared to how it used to be. And traditionally, the focus was on bones and teeth, yeah, because they protect the DNA better.
Right, bone and teeth are still the go-to sources. But the field is definitely pushing beyond that. The sources talk about a lot of effort going into improving techniques to get DNA from trickier materials.
Like what?
Things like decayed plant remains, dental calculus, that's the hardened plaque on teeth.
Oh, right.
Yeah, it can trap DNA from the host, their food, even microbes in their mouth. Also things like ancient parchments and even coprolites.
Coprolites. Fossilized poop. Wow. Okay, but getting DNA from something like that, or parchment, how do you make sure it's genuinely ancient and not just contamination? That seems tricky.
It's hugely tricky. Authenticating the DNA, especially from these unusual or poorly preserved sources, is absolutely critical. The sources really stress the need for what they call unified protocols.
Your name.
Basically, standardized methods agreed upon across different labs. Ways to rigorously test if the DNA is authentic, if it has the expected patterns of damage from being old, and how to minimize and account for modern contamination.
And speaking of the samples themselves, especially those museum specimens, there's a tension between getting the DNA out and preserving the actual object.
Real, definitely. That's the whole destructive versus non-destructive sampling issue. Historically, getting enough DNA often meant, well, drilling or grinding up a piece of bone or tooth.
Which museums are understandably hesitant about.
Exactly. These are often unique, irreplaceable specimens. So there's a big push to develop and refine non-destructive methods, or at least minimally invasive ones. Techniques for sampling bone surfaces, teeth without drilling deep, even getting DNA from things like insects trapped in amber, shells, or parchment without visible damage.
Sounds like they're constantly trying to get more information from less material.
That's the hope. The future tech will let us learn even more from tinier and tinier samples. But, despite all this amazing progress, the sources are also very clear. The field is still, quote, rife with challenges and pitfalls.
Okay, let's dive into those hurdles. What are the really big obstacles they face?
Well, the number one challenge is just DNA preservation itself. See, in a living thing, your cells are constantly repairing DNA damage.
Right.
After death, that repair machinery stops. So the DNA is just left vulnerable. Enzymes from the body itself, microbes in the environment, they start breaking it down.
It just decays.
Pretty much. And how fast depends hugely on the environment. Really cold like permafrost or very dry conditions like deserts. They slow down that degradation process. More of the original organism's DNA, the endogenous DNA, survives.
But warmer, wetter places are bad news.
Much worse. And even in good conditions, over thousands or tens of thousands of years, chemical processes, things like hydrolysis, oxidation, they just naturally fragment and damage the DNA strands.
And then there's contamination. That must be a nightmare, dealing with tiny amounts of ancient, damaged DNA.
It's arguably the biggest headache. Ancient DNA is present in such minute quantities, and it's often broken into tiny pieces. It's incredibly easy for it to be swamped by modern DNA.
From who? Like the people handling the samples?
Exactly. From archaeologists excavating it, museum curators handling it, lab technicians analyzing it. Even just dust in the air can carry modern human DNA.
So how do they deal with that?
It requires these incredibly specialized clean labs. Think positive air pressure, so air only flows out, not in. UV irradiation to destroy stray DNA. Constant bleaching and cleaning, filtered air systems. Full body suits for the researchers.
Wow. Sounds intense. And expensive.
Very expensive to build and maintain, which is one reason why there still aren't actually that many dedicated ancient DNA labs around the world relative to other types of labs.
And if you do get contamination or the DNA is super damaged, does that mess up the analysis, the fancy statistics they use?
Oh, absolutely. It complicates everything. High levels of contamination or severe damage can limit the types of statistical analyses you can confidently run. Sometimes researchers might only be able to reliably analyze the DNA fragments that show specific chemical signatures of ancient damage, like certain types of base changes at the ends of the molecules. That reduces the amount of usable data.
And this has led to some skepticism about the field in the past.
Yeah, the sources mentioned that some early, really bold claims about getting DNA from, say, insects in amber millions of years old, or dinosaur bones. Those were later shown to be contamination. That history means the field has had to work really hard to establish rigorous standards.
So how do they build confidence now? How do they convince themselves and others that their results are real?
It comes down to rigorous methods and quantitative analysis. They have to carefully model and estimate the levels of contamination in their samples. They look for those characteristic ancient DNA damage patterns. They use sophisticated statistical methods to filter out potential contaminants and authenticate the ancient signal. It requires a huge amount of careful work and computational analysis.
Okay, so it's challenging, but they're developing ways to tackle it. Let's shift gears now to the amazing stories this research is revealing, especially about human history, like mapping ancient migrations. The sources seem to have a lot on the peopling of the Americas.
Yeah, this is an area where ancient DNA has just completely transformed our understanding. The genetic evidence, overwhelmingly, points to the ancestors of the first Native Americans coming from populations in Siberia.
Okay, the Siberian origin seems pretty solid, but the sources mention some debate about the details, like did they hang out on the Bering Land Bridge for a long time, isolated, or was it a quicker migration through?
That's still being refined, and genetics is adding important details. For example, some studies looking at genetic markers like microsatellites or specific Y chromosome lineages passed down the male line suggest that certain indigenous populations, particularly in South America, have been relatively isolated genetically since the initial peopling of their specific regions.
But not all indigenous groups fit that exact same deep, ancient pattern.
That's right. Groups like the Nadine speakers in North America or the Inuit and other native Alaskan populations. Genetically, they share some deep ancestry with other indigenous Americans, like a common Y chromosome type called QM242. But they also have distinct patterns in their mitochondrial DNA from the maternal line and their overall autosomal DNA. These genetic differences, which line up pretty well with linguistic studies and even older studies on things like ABO blood group distributions mentioned in the source, they strongly suggest these northern groups represent later migrations into the Americas, specifically into the Arctic and subarctic.
So the genetics are showing a more complex picture than just one single arrival. Multiple waves, maybe.
Exactly. At least two, maybe more distinct streams of people coming in at different times.
And what about digging even deeper? Where did that first founding population actually originate from within Asia?
This is one of those really cool aha moments from the sources. Big studies using autosomal DNA, the bulk of our genome, particularly ones published around 2015 and 2018, they confirmed the Siberian link. But they also picked up this really faint, unexpected genetic signal, mainly in some Amazonian populations.
This signal suggesting what?
Suggesting some kind of shared ancestry with populations way over in Australasia, like indigenous Australians or Melanesians.
Whoa, hang on. Shared ancestry with Australasians in the Amazon? That sounds wild. How does ancient DNA explain that? Direct migration across the Pacific.
Huh. No, not quite that wild. The sources clarify this finding. It wasn't about people sailing from Australia to the Amazon. Instead, the signal seems to be linked to a very, very ancient population structure back in East Asia. There might have been a deep East Asian population, maybe related to the ancient individual known as Tianyuan man, whose DNA has been sequenced that contributed ancestry both to the groups who would eventually become the first Americans and to a sister group that eventually populated Australasia and places like the Andaman Islands.
Ah, okay. So it's like a really, really distant cousin relationship stemming from a common ancestral population deep in East Asian prehistory, not a direct connection between the Americas and Australia.
Exactly. It's a signal of shared deep ancestry from tens of thousands of years ago, revealed by subtle patterns in the genomes. Still a really surprising find.
Absolutely. Can the sources give us a rough timeline for these migrations into the Americas?
Yeah, that 2018 Nature study laid out some key estimates. They suggested the main founding population ancestral to Native Americans diverged from other East Asian groups maybe around 36,000 years ago. Then, there seems to have been continued gene flow with Siberian groups until about 25,000 years ago. After that, this ancestral American group seems to have become genetically isolated, maybe trapped in Beringia by ice sheets, or perhaps just south of them starting around 22,000 years ago.
And the split within the Americas between the ancestors of northern and southern groups.
That happened significantly later. The genetic data points to a split around 16,000 to 17,500 years ago, likely as people started moving south down the coast or through an ice-free corridor as the glaciers retreated.
Fascinating. And there was even a mention of movement back to Siberia.
Yeah, intriguing evidence for some populations moving back north and west into Siberia after about 11,500 years ago. So it wasn't necessarily just a one-way street.
And what about those specific genetic markers people often hear about, like Y chromosome DNA and mitochondrial DNA? Do they tell the same basic story?
They really do. They act like independent lines of evidence converging on the same narrative. The Y chromosome lineages common in Native Americans, primarily haplogroups Q and a specific branch of C called C3b, point strongly towards a central Siberian origin.
Okay.
And mitochondrial DNA haplogroups, the common ones being A, B, C, D, and a rarer one called X, also show links back to populations in the Altai-Baikal region of Siberia and potentially coastal East Asia.
So multiple genetic systems, paternal, maternal, and the whole genome are all painting a consistent picture of Siberian origins via Beringia.
Pretty much. And this strong genetic consensus really makes alternative ideas, like the old Solutrean hypothesis suggesting a migration from Ice Age Europe across the Atlantic, and makes them highly unlikely. The genetic signatures just don't support it.
Right.
The sources even briefly touch on some really old skeletal finds, like the Luzia woman's skull from Brazil, suggesting that the picture might get even more complex as we hopefully get more ancient DNA from that really early period, before the peak of the last ice age, the LGM.
Okay, let's jump way forward in time now to the period after European colonization arrived in the Americas. What does genetics tell us about that era?
Well, genetically, that later period is defined by massive population mixing, or admixture. Especially in Latin America and the Caribbean, you see this complex blending of indigenous American, European, and West African ancestries resulting from colonization and the transatlantic slave trade.
And what about in the U.S.?
The sources point out that in the United States, the situation historically and legally around Native American identity is often more complex than just genetics. It's frequently tied to tribal citizenship, culture, kinship networks. This relates to, a complicated history involving things like interracial marriage, sometimes controversial concepts like blood quantum used to measure ancestry, issues around tribal disenrollment, and also the phenomenon the sources mention of people self-identifying with a tribe without documented lineage, sometimes called the Cherokee syndrome or pretendians, in discussions about identity and belonging. It's a very complex social and historical landscape.
But purely from a genetic perspective, was there a study showing recent changes?
Yes, a really striking one used whole exome sequencing that's looking at all the protein-coding genes comparing ancient indigenous American genomes to modern ones. They specifically looked at immune system genes.
And found what?
They found a significant drop in the frequency of a particular variant as an immune gene called HLA-DQA1, which is involved in recognizing pathogens. This drop happened relatively recently, estimated around 175 years ago.
Wow, that timing. Late 18th, early 19th century. That points directly towards the impact of European diseases, doesn't it?
Exactly. It's interpreted as a direct genetic signature of the devastating epidemics, like smallpox, that swept through indigenous populations after European contact. These diseases caused immense mortality and acted as a powerful selective force, changing the genetic makeup related to disease resistance in the surviving populations and their descendants. It's like a scar left in the genome by that historical trauma.
Incredible and tragic. Okay, let's travel across the globe now to another major zone of ancient interaction, the Silk Road. What genetic stories are buried there?
The Silk Road, that vast network connecting Europe and Asia for centuries. It wasn't just silk and spices moving along those routes, it was people and their genes. Genetic studies of populations living along the Silk Road today, and some ancient samples too, show really clear evidence of admixture mixing between people with broadly European or Near Eastern ancestry and people with East Asian ancestry, especially in Central Asia.
So a real melting pot.
Very much so. And this mixing created high levels of genetic diversity in the region. You can actually see a geographic gradient in the genetic variation, correlating broadly with language families too. It shifts from more European-like in the West to more Asian-like in the East.
Is it a smooth gradient all the way along?
Not entirely smooth. The sources highlight a kind of division. The Western Silk Road areas, places like Armenia, Georgia, Azerbaijan, their populations tend to cluster genetically closer to other European and Near Eastern groups. Whereas the Eastern Silk Road regions, Uzbekistan, Tajikistan, Kazakhstan, they cluster genetically closer to Central and East Asian populations. And what's really interesting is looking at the specific ancestry components. While most Silk Road populations share some amount of a common European genetic signature, the Western regions have higher proportions of Near Eastern ancestry, while the Eastern regions have, unsurprisingly, much higher Asian ancestry components. It suggests different layers of history and mixing in different places.
Could the genetics tell which way the genes were flowing? Who was mixing with whom?
Yes, they could model the direction and extent of gene flow. The analysis suggested that in the Western Silk Road, there wasn't a huge amount of incoming gene flow, maybe some into Azerbaijan. But the Eastern Silk Road, that was much more dynamic. Places like Uzbekistan and Kazakhstan showed really high levels of admixture from multiple sources. Tajikistan also received significant genetic input, particularly from Central and South Asia. And the directionality was interesting too. The models pointed towards a predominantly east-to-west flow of genes from East Asia into those Eastern Silk Road populations and only a little bit into Azerbaijan. Gene flow from Europe seemed to primarily affect the Western Silk Road regions like Georgia and Azerbaijan. It paints a picture of complex, multi-directional movements over a long period.
So genetics isn't just tracking big migrations, but these more complex, long-term interactions too. Now, beyond just who went where, ancient DNA is also amazing for seeing how humans adapted to different environments or pressures. Natural selection in action.
Absolutely. This is where we get some of the clearest glimpses of evolution shaping us. A classic example the sources discuss is diet specifically, lactase persistence.
The ability for adults to digest milk. We know about that, but what does ancient DNA add?
It adds crucial timing and context. We know the main genetic mutation allowing adults to digest lactose is in a region near the lactase gene itself, on the MCM6 gene. Ancient DNA studies across Europe show that this mutation only became common relatively recently.
How recently?
Well, thousands of years after the archaeological evidence shows people started dairy farming, raising cattle for milk.
That's surprising. You'd think if people started using milk, the gene allowing them to digest it would spread really quickly.
You might think so. But the ancient DNA suggests there was a significant lag. The strong positive selection favoring this lactase persistence allele seems to have really kicked in later and was maybe strongest primarily in Northern Europe, not everywhere that dairying was practiced early on. The reasons for the delay and the geographic focus are still debated. Maybe milk was mostly processed into lower lactose forms like cheese or yogurt initially, or maybe selection only became really strong under specific dietary or environmental stresses.
Fascinating. Okay, what about another visible trait, like skin color?
Pigmentation is another great example of recent adaptation revealed by ancient DNA. Genomes recovered from Upper Paleolithic and Mesolithic hunter-gatherers across Eurasia generally show they carry gene variants associated with darker skin pigmentation.
So earlier Europeans were darker skinned.
The genetic evidence points that way, yes. The specific gene variants that are strongly associated with lighter skin in modern Europeans. Alleles in genes like SLC24A5 and SLC45A2, which influence melanin production, appear to have entered European populations later. They likely arrived primarily with the migrations of early farmers from Anatolia during the Neolithic period, starting around 8,000 years ago.
And then natural selection favored these light skin variants, perhaps linked to vitamin D production in northern latitudes.
That's the leading hypothesis. These introduced light skin alleles were strongly selected for over the following millennia, leading to a gradual depigmentation of European populations during the Holocene. The sources note that the specific combination of pigmentation alleles common in many Western Europeans today is actually quite recent, likely only reaching high frequencies in the last 5,000 years or so.
Wow. So these traits we might think of as ancient are actually quite recent evolutionary developments. What about the immune system? That must be under constant pressure from diseases.
For sure. Immune system genes show really complex patterns of selection. Ancient DNA studies are uncovering selection acting on immune genes in Europe over the last 10,000 years. And we already talked about that HLA example in the Americas, showing selection pressure changing after European contact introduced new diseases. And here's where it gets really powerful. Sometimes scientists can recover DNA from the ancient pathogens themselves alongside the human DNA. One source mentioned recovering DNA from Yersinia pestis, the plague bacterium, from Bronze Age individuals.
So you can see the pathogen and the human genetic changes side by side.
Exactly. You could potentially correlate specific changes in human immune genes directly with the presence of particular diseases in the past. It's like watching the evolutionary arms race play out in ancient history.
That is incredible. Speaking of disease resistance, the sources had a really detailed example about malaria, didn't they, in Pakistan?
Yes, a study on the Makrani people of coastal Pakistan. They have known ancestry tracing back to the Indian Ocean slave trade, bringing people from Africa to the region. Researchers looked at a gene called DARK. A specific variant of this gene results in the Duffy null blood group phenotype. The key thing is, this Duffy null variant provides strong resistance against infection by Plasmodium vivax, one of the main malaria parasites.
And this variant is common in parts of Africa where malaria is endemic, right?
Exactly. So the researchers looked at its frequency in the Macrones. They found the Duffy null allele at a frequency of around 50%.
Okay, and how does that compare to their overall African ancestry?
That was the crucial part. Based on genome-wide estimates, their overall African genetic ancestry component was around 30%. The fact that the Duffy null allele frequency, 50%, was significantly higher than their average African ancestry, 30%, is strong evidence for positive selection. It means that after this resistance allele was introduced into the Macrani population via gene flow from Africa, it was strongly favored by natural selection, likely because P. vivax malaria was and is a significant health threat in that region. Its frequency increased well beyond what you'd expect just from the initial admixture event.
So it's a clear genetic footprint of both historical migration, the slave trade, and ongoing evolutionary pressure, malaria.
Precisely. A really powerful example combining history, genetics, and adaptation.
The sources also had a quick mention, almost an aside, about adult male height correlating with the amount of ancestry derived from ancient Pontic-Caspian steppe pastoralists.
Right, just another hint at how genetic components tracing back to specific ancient populations could be statistically linked, sometimes controversially, to complex traits like height in modern populations.
Okay, one last major area before we wrap up. Ancient DNA isn't just about us humans or even wild animals. It's shedding light on how humans shape the world around them, especially through domestication. There was that fascinating potato story.
Yes, the Four Corners potato, Solanum jamesi. It's a wild potato species native to parts of the U.S. Southwest, mainly Arizona and New Mexico. But archaeologists had found remnants of these potatoes at ancient sites well outside that natural range, up on the Colorado Plateau, in areas inhabited by the ancestors of modern Pueblo, Diné, Navajo, Southern Paiute, and Apache peoples.
So the question was, how did they get there?
Exactly. The genetic study analyzed modern and ancient potato samples. They found that the potatoes growing near these archaeological sites, sometimes in isolated patches, were genetically distinct from the main populations down in the species' natural range along the Mogollon Rim.
Suggesting people moved them.
That's the interpretation. The genetic patterns strongly suggest that ancient indigenous peoples were actively collecting these tubers, transporting them hundreds of kilometers, cultivating them, and trading them across the Colorado Plateau for millennia. The researchers see this as clear evidence of human management, transport, and likely the very early stages of domestication, deliberately propagating a useful plant outside its wild habitat.
And they called it an enduring ecological legacy.
Yes, highlighting how indigenous peoples were sophisticated land managers and agricultural innovators who actively shaped the plant life of the Southwest long before European arrival.
And this potato wasn't just any plant, right? It's quite nutritious.
Apparently so. Yeah, highly nutritious, and could have been a really valuable food source and trade item, maybe a lost sister crop, alongside the famous maize, beans, and squash.
And didn't the source say local tribal members already suspected this connection?
It did mention that, yeah. That tribal knowledge hinted at a long history of human interaction with this potato, which the genetic study then scientifically confirmed. It's a nice example of how scientific research can sometimes align with and validate traditional ecological knowledge.
A great story of human ingenuity shaping the landscape. And they're planning more research.
Yeah, further genetic work to understand the domestication process even better shows how these studies often open up more questions.
Okay, before we finish, the sources did briefly touch on the sort of ethics and global context of this research, too.
Right. They pointed out that historically, a lot of the ancient DNA labs and funding have been concentrated in Europe and North America, the global north. This has sometimes led to practices described as helicopter research or parachute science,
where researchers from wealthier countries fly into places in the global south, collect samples, sometimes sensitive ones like human remains, analyze them back home, and it may be published without much collaboration or involvement from local scientists or communities.
That's the concern raised in the sources, yeah. It brings up important ethical issues about equitable partnerships, sharing benefits, respecting local beliefs and regulations regarding ancestral remains, and building research capacity within the regions where the discoveries are being made. The field is definitely grappling with these issues more openly now. And related to that, as aDNA research moves into more recent historical periods studying population changes over the last few centuries, for instance, geneticists are realizing they need to collaborate much more closely with historians, just like they've learned to work with archaeologists and anthropologists for deep prehistory. Understanding the historical context becomes absolutely crucial.
Well, we have covered an incredible amount of ground today, geographically, chronologically. It's amazing.
It really is.
From tracing those first epic journeys into continents, to seeing evolution literally changing our genes in response to disease, uncovering lost agricultural practices, mapping out the family trees of extinct giants, ancient DNA is just such a powerful window onto the past.
It provides a level of detail and sometimes certainty about past events and relationships that was just unimaginable not that long ago.
It really makes you stop and think, doesn't it?
Mm-hmm.
What echoes of these ancient migrations, these adaptations, these millennia of interacting with the environment and each other, what echoes are still resonating within our own DNA today? What other incredible stories are still locked away in those ancient fragments, just waiting for the right techniques, the right questions to bring them to light?
It definitely reminds you that history isn't just something in books. It's written into us, into the living world.
Absolutely. And if this journey into our shared genetic past, this unfolding story of life, fascinates you as much as it does us, then you absolutely belong here with the All Sorts podcast community.
Yeah, come join us.
Join us as we keep exploring these incredible frontiers of knowledge. The very best way to make sure you catch every single deep dive into topics like this is to hit that like button, share this episode with someone else who's curious, and definitely subscribe to the All Sorts podcast on YouTube.
Please do.
And find us on X, Instagram, and Threads too. Stay part of the conversation. We've already got a whole library of other deep dives covering all sorts of fascinating subjects waiting for you to explore. Thank you so much for joining us for this deep dive on the All Sorts podcast.